Greetings, ladies and gentlemen, and welcome once again to the Compendium of Mad Science. Yes, today is Microscopy Monday. We're going to be looking at the Hematoxylin and Eosin stain, H&E stain. It's a very common, very basic stain. Involves a couple of steps, but they're fairly simple. I'm going to walk you through them. In the previous Microscopy Monday, I showed you how to prepare some slides. I've actually made a repeat of that with some cheek cells, and we're going to be going through a couple of extra steps to stain them. This I'll show you before and after staining, just so you can see the full effect. Right, let us begin. Right, so to perform our H&E stain, we prepare a couple of slides, just as we did last week. With one difference: once you've made the smear, you put it... Now first of all, you need to make sure that the sample, which is on top, does not come into contact with the hot air directly. So what I'm going to do is I'm going to put this over the top of this for 10 seconds. So I've taken the samples, just as we did before, and I've air dried them. After that, as you can see, I've just performed fixation, or fixing, which chemically bonds the proteins on the cells to the surface of the slide, so that we can wash and do stuff without making a huge mess. So once we've fixed all three of our slides, we can begin the staining process. There's a lot to staining that I'm not going to cover here, simply because it's so complex. Right, so first up, I'm going to take our slides. So we have two smears that we used, just like I showed you last week. This smear here is a much bigger sample. This is similar to what you do with a blood smear. I'll show you how to do that on the next Microscopy Monday, when we actually do a blood smear. But in the meantime, suffice it to say, it's a very big sample with a higher density over here than on this side. So first up, we just put a couple of drops of this onto our sample. Now what I'm going to do is something you probably shouldn't do yourself. So I'm going to smear it out a bit. The reason you don't usually do this is because, as you can probably see, it's causing quite a bit of cross-contamination between the slides. And I've actually just noticed that these aren't spreading out very well, so I'm just going to redo them. This one's looking fine though. Right, so we need to leave this for about 10 minutes. Now that we've left that for 10 minutes, during that time I've actually made up a quick control, or rather I took last week's control, this is actually last week's slide, and I've fixed it. I've also taken the liberty of putting down some greaseproof paper, because I had the sudden recollection that this very badly stains plastic if you put it on it, and my desk has a plastic anti-static mat on it. So what I'm going to do next is I'm just going to take these and run them through the rinse. Again, I'm going to leave the top wet and dry the bottom. You can flip them above the heat gun if you really want, but I wouldn't advise it. So what we're doing here is we're just washing off the excess stain, ready for the next stage, which is the eosin stain. Again, we're just going to do exactly what we did last time. By the way, this stuff stains your clothes bright orange. Make sure you're wearing a lab coat and gloves, because it also stains your skin bright orange. And trust me when I say you won't like it. And again, I'm going to do this for 10 minutes. Right, so that's been 10 minutes. So again, we just wash them off just as we did before. Oh, look at that colour. this last one's going to be good. Oh. Got a nice strong stain on it, which may mean that the sample's actually too dense to use properly. Hopefully that won't be the case, but with any luck we'll be able to see quite a lot with that one. Right, so again I'm just going to leave them for a bit to dry out. Right, so let's look at that on the camera. So this is the control slide with no stains applied. You see here we've got a single epithelial cell and, oh, what's that? Looks like a... That just a wee fibre, probably from my clothing. Here we have a single cheek epithelial cell. Let's just zoom in on that. Focus. You can make out the nucleus, but it's difficult to make out anything else. The contrast isn't particularly good. Zooming out, you can just see a bunch of cell debris. It's not terribly, the contrast isn't terribly great. Moving on to the first of our stained smears. This is the smear 1. And you can see right away that it's easier to spot the cells and the cell debris as it is stained purple or pink. Zooming in on this group of cells here, you can see that the nuclei have stained violet while the rest of the cell has stained purple or pink. It may also be possible to make out a few of the cell organelles, but I'm not entirely certain of this. It may just be debris on the slide or on the microscope. You can see the cell's actually folded over in itself a little bit. The cheek epithelial cells are fairly flat, if I recall correctly. Amorphous layers above a more linear organised layer. Moving on to the next of the stained slides, we again see that so this is the second of the stained smears. The second slide here isn't too great. There's a... I'm not sure if I've overheated it, but a lot of the cells seem to be badly denatured. And in some cases outright destroyed, as you can see, like that group in the bottom there. Right. Yeah, in fact, zooming in on this one, you can see it's quite it's very badly dried out, so it's probably what's happened is when I made up the slide, I've not given it enough time to air dry. So when I've heated it, the cells have burst and then stuck down. This is the densest of the 3 slides we prepared, and as you can see straight away there's a huge difference in this slide. Incidentally, these tears on this little film on the bottom here is caused by my terrible habit of smearing out the stain. This is the main reason you don't smear out the stain like this, because you'll damage your sample. This little skin formed on the slide is actually a bunch of proteins from my mouth. Some of them are used in saliva, others are part of the cell which escaped when I was chewing on my cheek to get the sample. As a result, you can see there's quite a few displaced, disembodied nuclei lying around. Some of these may in fact be other microorganisms, but I'm not entirely certain about that. I'd be more inclined to believe that they're all nuclei rather than a few being microorganisms. Having said that, Your mouth does have a microfauna and flora, microflora and fauna. Your mouth is a microenvironment, so there's an entire microcosm of little organisms living in it at all times. So some of these are in fact bacteria. A gram stain would probably allow us to differentiate between human cells and bacterial cells. These here rather remind me of flagellates. They look like they've got a nucleus. They look like they've got a nucleus, but I'm not entirely sure. I hope that they are, in fact, debris from epithelial cheek cells. But they kind of remind me of filarial nematodes, or, as I mentioned, flagellates there. I rather hope that's not the case, though, because I'd rather not have that living in my mouth. And this here is Cthulhu, or Cthulhu's microscopic cousin. Again, that's just folded, a little skin along the bottom that's been folded over. Replace the batteries on the lamp I was using. Oh, that's much better. Yeah, I'm pretty sure those are disembodied nuclei. Again, we overheated this sample. I didn't think it was dry when I heat fixed it, but I can tell you right now it definitely wasn't. In fact, there's a good example there. That's a group of cells which have burst, and that's just their flattened remains stuck to the slide. It's like something out of a horror film. Again, you can see quite a large concentration of cells in this area. There's a bunch of debris again. Again, you can see the outline of several cells here, which makes me think that those were indeed cell fragments which have burst open, and we're just seeing the debris. There's a couple of disembodied nuclei there as well, so that lends credence to that hypothesis, but again, I might be wrong. If anyone recognises these little crescents and sort of fractal shapes, just let me know in the comments, it'd be useful to know. You generally shouldn't make a smear this densely packed, for the simple reason that it means there's a lot of stuff to look at, and it's difficult to spot any particularly interesting features because there's just so many to look at. There's a lot of debris around here. And again, there's a lot of damage to this slide. Which is entirely my own fault, of course, but... There's a couple of good samples around there I should probably have zoomed in on. Pretty sure the ×10 objective lens on my microscope, sorry, the ×20 objective lens on my microscope is actually out of alignment with the other two objective lenses. I'll need to see about getting that fixed. Hopefully it's not serious damage, but it is a bit of a nuisance when I try and center something, zoom into the next level and then center it on it again. Yeah, you can see here this sample's probably not dried out entirely before I heat fixed it. See, it's burst in two places and the cell remnants have stuck to the slide. Well, ladies and gentlemen, I think that will fulfill our mad science quota for the day, and so concludes another Microscopy Monday. On the next Microscopy Monday, we'll be looking at a blood smear and how to perform it and what it looks like and what blood cells look like under the microscope. So this will be using my own blood. Next week we'll be returning with a Technical Tuesday. Hopefully we'll be back on our original schedule. I'm planning to return. I'm planning on changing the schedule a little bit. I'm planning on actually filming Microscopy Monday on a Monday and Technical Tuesday on a Tuesday. And I'm going to be adding a new segment called Scientific Saturday where we ramble. Again, we'll still be doing the alternating week thing. So one week will be Microscopy Monday, the next week Technical Tuesday, the next week Scientific Saturday. I'm not entirely sure what day I'll be releasing these on. It'll probably be the Sunday, hopefully. Maybe Monday. We'll see. We'll see. I'm not planning on sticking to a perfectly rigid schedule, but anyway, until then, folks, have a nice evening. This has been the Compendium of Mad Science. This was another Microscopy Monday, and I am your resident mad scientist, Callum P. Robertson. Good night, folks.